All right. Welcome, everyone. Thank you for joining us for today's GemPharmatech webinar for January 2022, getting a new start in the new year. So my name is Jing Li. I'm the Director of Life Science and Technology at GemPharmatech US. I'm introducing my colleague, Dr. Steve Smith, who is our Director in Business Development, also in the US team, to give us a discussion on our flagship program, KOAP, Knockout O project. So before Steve dive into the details, here is a brief intro to Steve. So Steve completed his undergraduate study at Beloit College before conducting his graduate work at North Carolina State University in the Department of Microbiology, where he studied lipid biochemistry in Baker's East in the laboratory of Leo Parks. He went on to Gladstone Institute of Cardiovascular Disease in San Francisco to conduct a postdoctoral fellowship in the laboratory of Dr. Robert Fares, studying genetic underpinning of cholesterol metabolism and triglyceride synthesis. He began his industry career at Parke Davis Pharmaceuticals, where he studied diabetes and glucose metabolism. He joined GemPharmatech as our BD director last year, and his goal is to increase our corporate exposure to the US market. So without further ado, Steve, take it away.
Thank you for that very nice introduction, Jing, and thank all of you for joining us today and learning a bit more about GemPharmatech and our flagship opportunity for the KOAP, or Knockout All project, and all of our genetically modified mouse models and various applications in research.
Audience, if you have questions, feel free to use the chat or Q&A to send it in as Steve go along with the delivery, and then we'll come back to your questions or discussion points after the webinar.
Yes, thank you very much. So with GemPharmatech and creating this very large library of genetically modified mouse models, I want to mention that lab animals have been a huge source of for research animals, research community in academia and industry, and with their research with the animal models, they've resulted in quite a few Nobel Prizes in medicine and physiology. Mouse models are a great resource, again, for academic and industry research. They're a very close genetic relationship with humans, very highly studied, easy animal to work with for research, particularly owing to the knowledge of their own genome and genetic makeup. So very clear genetic background and annotation, very small body size. They grow very rapidly, reproduce very rapidly. So you can obtain enough animals to have a statistically significant research study and conduct follow-up studies and be able to conduct it in the convenience of a vivarium at a research institution, academic or industry. There are a number of ways to obtain mouse models, either spontaneous mutants or random induced mutations, can be induced through radiation, ENU, gene trap, and that's considered forward genetics. Also, conducting reverse genetics allows very targeted gene modifications, and for modifications, whether they're a point mutation, a knock-in, or a knockout, can be done using ES cells for targeted mutagenesis or endonuclease-mediated mutagenesis. The development of these technologies has resulted, as I mentioned, a number of Nobel Prizes, going from early 1950s with Watson and Crick discovering of the DNA double helix, moving forward in ES cell-based homologous recombination in the very early 1980s. About 15 years later with zinc finger technology, then TALEN, and now CRISPR. It's now 10 years ago, where CRISPR came forward as enormous opportunity for rapid generation of transgenic knock-in and knockout animal models. With CRISPR-Cas9 allows the production of very specific mutations or alterations in the genome through the CRISPR and Cas9 recombination. Some of the advantages, primary advantages of CRISPR-Cas9 versus ES cell targeting, which had been the state of the art prior to CRISPR-Cas9. ES cells, roughly 14, 16 weeks just to get the knockout clone in ES cells, then injection into blastocysts, and then another 24 weeks before the animals are on the ground. With CRISPR-Cas9, using the knowledge of the mouse genome and any mutation that you would like to introduce, takes roughly 2 to 6 weeks, then the 24 weeks of generating heterozygous animals. So cutting out roughly 10 to 14 weeks just from the development of the animal model. And time is money, time is research time. And so being able to reduce that is critical. And moving into the genome modification strategy, this technology, CRISPR-Cas9, allows generation of standard and conditional knockouts, knock-ins, complex modifications, and transgenic models. Another advantage with CRISPR-Cas9 is that you can generate these models in a wide range of mouse strain backgrounds: Black6, BALB/Cs, NODs, the NCGs, immune incompetent, FVBs, 129s, and also generate models in the rat genetic background, the SD background. ES cell technology was pretty limited to just 1 or 2 genetic backgrounds. In generating a conventional gene knockout using the Cas9 system, the endonucleases are designed for introduction of or deletion of a region of interest. So in this case, want to remove exon 2. So through the gene editing, create the exon deletion and introduce a frame shift, or you can introduce a frame shift codon, or you can do a complete knockout depending on where your CRISPR-Cas9 endonuclease sites are binding. Also, when generating a conditional knockout, this is essential when a standard knockout leads to embryonic lethality or other lethalities or toxicity in animals, rendering them impossible to study with a knockout. With the knockouts, with these conditional knockouts, then it's very valuable for looking at the knockout in a specific tissue or organ, and looking at secondary effects and monitoring functions of during development at various stages, particularly if a conditional Cre line is used. With this model, LOXP sites are introduced in the genome around the exon or area of the genome that is desired to be deleted. Then when Cre is present, it knocks out just that region between the LOXP sites resulting in deletion of gene function or altered gene function. And so with these applications, you could use a tissue-specific Cre driver to drive the Cre expression just at particular times or in tissues of interest or cells of interest. And when these animals are crossed with the floxed allele, animals with the floxed allele, they result in the inactivated gene. As an example, the PDK1 animal model is found that when a homologous knockout is trying to be generated, it's found that no homologous knockout animals are born because it's an embryonic lethality. And if it's desired to study PDK1 just in tendon development, it can be crossed with a tendon-specific Cre driver animal and resulting in homologous knockout only in tendons for that study. If it's desired to study PDK1 only in cardiac development, again, the embryonic lethality, if it's a total knockout, but when using a Cre driver, inducible Cre, can knock out the gene, PDK1 gene, only in the cardiac muscle at a particular time in the animal's life after tamoxifen induction. Knock-ins, generating knock-ins using Cas9, again, there is, can do looking at the donor, again with the CRISPR-Cas9, can do induction to have a gene open reading frame just upstream of the first exon. And in this case, it'd be disrupting the endogenous gene, or if you wanted to maintain the gene of interest and have your open reading frame of interest just downstream of your gene, have that tied in open reading frame with the existing gene. When making transgenes, in the standard back engineering process, the transgene is made up on and then released from the plasmid, and this is just injected into embryos for transgene uptake and expression. However, with CRISPR-Cas9, using this technology and having it targeted to a safe harbor locus, such as H11 or ROSA 26, this same through the CRISPR-Cas9, this all can be incorporated into H11, ROSA 26, and have much more stable expression over time in the animals. Whereas with the random insertion, it may inactivate a gene in the genome, resulting in unstable expression. Point mutants can also be induced using CRISPR-Cas9. In this example, a point mutant within exon 2 is introduced with the CRISPR-Cas9 targets, and now with the mutant allele. And this is very, very important because in human disease, a huge number of human genetic diseases are caused by single point mutations. There's about 25% of human diseases that are resulting from a actual deletion, significant deletion in the gene, but well over 50% are caused by single point mutations. So another big advantage with CRISPR-Cas9 is that the number of pups and the rate of the models going germline are far higher than working with ES cell technology. With the Knockout All project, our goal is to knock out all genes in the mouse genome by 2023. We're well on our way, and that has resulted in GemPharmatech having the largest gem resource library in the world, with over 9,000 knockout lines, over 8,000 conditional knockout lines already, and more in the works. So over 6,000 a year are being generated. GemPharmatech also has software for accurately generating these models as well. So they're with our technology allows very rapid identification of the CRISPR-Cas9 target sequences and knowing where in the genome to introduce mutations or deletions. With the genes that are being deleted, or the knockouts and conditional knockouts being made, going through all of the various gene families, knocking out genes in subcellular regions. And again, we're tracking all of those. Also, these knockouts naturally then fall into all tissues and organs. And within our website, you can follow, not only look up just the gene or the strain of interest, but you can learn about the pathways that are involved. And as I said, learn about the pathways involved, and there's you can look up on our the bar at the top of the page and look up genes and knockouts that are involved in Alzheimer's disease, angiogenesis, and other disease areas. For the conditional knockout strains, these need to be crossed with a with a Cre model, and resulting in the FLOX allele being deleted, whether it's in nervous tissue, the cardiovascular system, digestive system, immune system, and many others, very specific tissues and cell types. And over 200 Cre strains are available. With the Cre Expression Analysis, this can be utilized for downstream fluorescence analysis to have a fluorophore that is expressed only in particular tissues for immunohistology, immunofluorescence, and can be set up with FLOX, then fluorophore FLOX, and have this be deleted when it's crossed with a particular Cre strain. An example of that here is with the PDX1 P2A Cre, that when Cre is not present, expressed in certain cells, and after Cre expression, Cre then results in TD tomato being expressed. With all these models being generated, GemPharmatech naturally has very high QC standards to be certain that the knockout of interest is being made as desired. With our knockouts, there's well over 100 publications that have come out using our KOAP models, and this number is growing by the day, by the issue of the journal that comes out. With these KOAP models, there's a number that we have made that are specifically modeling human diseases. An example here is Duchenne's muscular dystrophy, or DMD. this mutation in humans causes a truncation in the dystrophin protein in skeletal muscle and cardiomyocytes. It's a rare disease. It's an X-linked gene, and so we have generated a mouse model of DMD. There are a couple of therapeutics out on the market, and there are more in the works now with using CRISPR-Cas9 technology. And with our animal model, modeling human disease, you can see that as following the grip strength in the animals, you can see that as the animals age, after age 16 to 20, they have statistically reduced grip strength compared to wild type animals. And that's also manifest and observed over time with rotorod test results. And looking at CK levels, you can see that in the DMD animals, there's CK that indicating the muscle depletion, muscle injury over time does come out as the animals age, that from 4 to 5 weeks of age, they're all similar, but the DMD have elevated CK, and by 22 weeks of age is markedly increased. And this is also seen in the histology analysis, just H&E staining. Another model looking at hemophilia. And hemophilia, again, it's a rare disease in humans. There's two varieties. There's hemophilia A, caused by a mutation in the F8, and hemophilia B, caused by a mutation in F9. Hemophilia A, the preponderance of patients, another X-linked, another recessive rare disease. There's a number of therapeutics out on the market. Two that are in phase 3, others that are still earlier in the pipeline. We have a mouse model recapitulating the human disease of the hemophilia A with introducing a mutation in exon number 16. And you can see that in this model that the factor 8 is dramatically reduced in the animals, in both the females and the males, resulting in dramatically increased bleeding weight or bleeding volume, the time the animals are bleeding, and the amount of red blood cells that are observed. GemPharmatech has also been using the Cas9 technology, CRISPR-Cas9, to generate some humanized models. And humanized models are just that. They're a way of studying human disease more accurately in the mouse. And with gene humanization strategies, can either approach it by replacing the entire murine gene, so whole gene humanization, replacing it with the human gene, and whether it's replacing it or having it expressed in tandem, or introducing just a partial gene humanization, which might just include the extracellular domain or ligand binding domain. With this, GemPharmatech has generated over 700 humanized strains, many for humanized immune checkpoints, and doing for the humanized checkpoints, generating the models in both the BLAC6 and the BALB/C backgrounds. Shown, and with this then for humanized checkpoints, single target, double, triple, and quadruple target animals to best recapitulate human disease and helping to understand the potential activity of chemotherapeutics in mice. As an example, using the chemotherapeutic, being an antibody from Roche against the TIGIT protein. You can see that when working with an animal model in the BALB/c, the tumors grow very rapidly. The CT26 tumors grow rapidly in the BALB/c background, but the antibody therapeutic works very well in reducing tumor volume. However, because of differences in the immune system and immune response, when working with a similar MC38 tumor line in the BLAC6 background with the animals containing humanized PD-1, human TIGIT, that this does not result in tumor regression after or during treatment. And that circles back to the differences in the immune response and immune genes in the BLAC6 background versus the BALB/c background. Looking at changes in natural killer cells and Tregs and their response to the tiragolumab treatment, you can see that in the BLAC6 animals, there's a slight increase after treatment, slight increase in Tregs and NK cells. In the BALB/c background, there's a much more dramatic increase in NK cells and repression in the Tregs. GemPharmatech has also generated 5 humanized ACE2 knock-ins for the study of COVID-19 infection and developing therapeutics against COVID-19 while using mice as the animal model. With these models, just following the expression or following the infection BLAC6 with a humanized ACE full-length ACE2, you can see that there's expression very high in the heart, kidneys, lungs, and the animals are healthy during the infection and following infection and clearance of infection. And these animals have been used in published research. Here, a paper in Science that came out in the past year showing utility of this animal model in following, in learning about new chemotherapeutics against ACE2 or against COVID using the humanized ACE2. This just shows the huge outreach and expansion of the mouse genome modified mouse models with over 78,000 strains globally through MMRRC, Taconic, Riken, JAX, and GemPharmatech. GemPharmatech serves as a one-stop solution for biomedical research. So not just our KOAP library, but we also have, we can conduct breeding, cryopreservation, conduct contract research, preclinical efficacy and phenotyping services, and we ship animals around the world. So thank you very much for attending today, and I look forward to any questions you may have.
Well, thanks, Steve. And I have been monitoring the Q&A and chat. We did receive a couple of questions, so I'll get the question started and going your way. So the first question is, can you comment on your QC processes to ensure that your constitutive or conditional knockouts block the transcription and translation in all or the targeted tissue, respectively?
QC is primarily going back to sequencing the regions where that are designed to be knocked out, and whether it's the exons are deleted or the LOXP sites are introduced in a conditional knockout. That's the first and primary level of QC with the animals as they're generated.
Yep. So add on to what Steve shared, we also do off-target analysis, and we can also do custom required off-target analysis if you want to increase,
Yes.
like look at more potential sites. Second question, can you talk about, you mentioned humanized animals. When you do the humanized animals, do you target your strategy on diseases primarily, or you're doing in a more broad way of humanizing every gene in the mouse?
It's primarily, when introducing a mutation, is primarily for accurately modeling a human disease. For the humanized immune checkpoints, that's a very targeted approach for very specific applications, those models.
Great. We got a question asking about, well, first of all, great presentation. Could you please provide some insight into delivery timelines for a KO-KI or a CKO-KI model?
Typically, it's from introducing, from beginning this project until delivering the animals. It's roughly 6 months, but it might be a month or two longer depending on the complexity of the model and the desired knockout or knock-in that's introduced.
All right, that's on our custom model generation pipeline, right?
Yeah. Yes.
So if you are ordering a KOAP line, the timeline is much shorter. They are pretty much either on the shelf being live or in stored as frozen material, sperm or embryo. They are one reservation away from being shipped. So that timeline typically is within a month or maximum 2 or 3 months.
Yes, just from reviving from frozen material to live animals, it could be in your vivarium within 2.5 to 3 months.
All right, yeah, you got a thank you from our audience who asked the question. One last question, as far as I can tell, will the slides be shared with the attendees?
Yes, they will be available. Our presentation should be available on our website.
Yeah, you can visit our website, is on demand.
Yes.
After sending your request, we'll provide it to you, not only the slides, but also the recording for Steve's presentation today.
Thank you very much. I appreciate your attendance. Bye-bye.
Bye-bye.